The cell cycle is a series of events involving the growth, replication, and division of eukaryotic cells, consisting of interphase, mitosis, and cytokinesis. There are three stages of interphase. They are G1, S, and G2. During G1, or the first gap phase, the cell grows in preparation for division. At the end of G1, the cell goes through the first checkpoint, where it will decide whether or not to proceed to division, or if it will go into a resting stage called G0. During the S phase, DNA is synthesized, and during the G2 phase, the cell continues to grow, synthesizes protein, and replicates organelles. At the end of G2, the cell goes through the second checkpoint, making sure that the DNA was replicated properly. After interphase, the cell enters mitosis, which is the process by which a eukaryotic cell divides into two identical daughter cells with the same number of chromosomes. Mitosis is responsible for the growth and repair of multicellular organisms, as well as asexual reproduction in organisms. The first stage of mitosis is prophase. In this phase, the nucleoli disappear, the nuclear membrane breaks down, and the chromatin condenses into visible chromosomes. Centrioles begin to migrate to opposite poles of the cell, and the meiotic spindles begin to appear. In metaphase, the chromosomes align on the metaphase plate, and the meiotic spindles extend from the centrioles and attach to the centromeres. At the end of metaphase, the cell goes through the third and final checkpoint, making sure the chromosomes properly aligned in the center of the cell and the spindle fibers properly connected to the centromeres. During anaphase, the centromeres holding the sister chromatids together break apart, the spindle fibers contract, and the sister chromatids separate, each becoming their own chromosome. In telophase, the chromosomes gather at opposite poles of the cell and start to form the nuclei of the two new cells. The nucleoli start to reappear, the nuclear envelope reforms, and the chromosomes unwind. The third part of the cell cycle following mitosis is cytokinesis, which accompanies telophase. The cell membrane pinches on both sides, forming the cleavage furrow. The cytoplasm and all the replicated organelles are split between the two new daughter cells, and the cleavage furrow pinches all the way in, separating the cells from each other. At the end of mitosis, there are the same number of chromosomes in the daughter cells as in the parent cell. In humans, 2n equals 46, and after mitosis, there are still 46 chromosomes. A similar but also very different process from mitosis is meiosis. Meiosis is the cell division occurring in sex cells where the two daughter cells have half the number of chromosomes as the parent cells. The function of mitosis is to produce two genetically identical cells, while the function of meiosis is to produce genetically unique haploid cells. In meiosis, the cell goes through two divisions opposed to one like in mitosis. This is what makes the daughter cells haploid instead of diploid. Meiosis promotes genetic diversity through crossing over, independent assortment, and random fertilization. Meiosis 1 separates homologous chromosomes, and meiosis 2 separates sister chromatids. During interphase, DNA is synthesized and the cell prepares to enter meiosis 1. During prophase 1, the nuclear envelope breaks down, the spindle fiber apparatus begins to form, and the chromatid condenses into visible chromosomes, made up of two sister chromatids bound by a centromere. Next, two homologous pairs of chromosomes bind to each other, forming a tetrad. This process is called synapsis. Then, sections of each chromosome are exchanged between non-sister chromatids. This is called crossing over. During metaphase 1, the tetrads align on the metaphase plate, the centrioles have migrated to opposite poles of the cell, and the spindle fibers connect to the kinetochore of each homologous chromosome. Then in anaphase 1, the spindle fibers shorten, pulling the homologous chromosomes to opposite ends of the cell. The sister chromatids of each chromosome stay connected to one another. In telophase 1 and cytokinesis, the cell splits into two haploid daughter cells. The nuclear envelope forms and the chromosomes decondense, preparing for meiosis 2. The first stage of meiosis 2 is prophase 2. The nuclear envelope breaks down, the chromatid condenses into chromosomes, and the spindle apparatus forms. In metaphase 2, the chromosomes align on the metaphase plate, and the spindle fibers attach to the kinetochore of each chromosome. In anaphase 2, the spindle fibers pull the sister chromatids apart to opposite poles of the cell. Now, each sister chromatid can be considered its own chromosome. The cell membrane begins to pinch, forming the cleavage furrow. In telophase 2, the nuclear envelope forms and chromosomes uncoil. Then cytokinesis splits the two cells, resulting in 4 genetically unique haploid cells. The 4 daughter cells have half the number of chromosomes as the parent cell. When the two gametes come together during sexual reproduction, they result in a diploid cell. The big difference between mitosis and meiosis is mitosis produces 2 identical diploid cells, while meiosis produces 4 genetically unique haploid cells.